All right, sponges are multicellular, but they don't have tissue development. So as we were talking about before, how we have endoderm, mesoderm, and ectoderm, sponges don't have that. That applied to tissue formation. And even though sponges are multicellular, they don't have their cells arranged into tissues. This is going to be the only phylum, the simplest of all the, the simplest of all the phylums, and they don't have tissue development. So they are multicellular, but the cells are working independent of each other. Now, sponges are sessile as adults. This is the type of feature in which the sponge itself is fixed to a reef. They can be fixed to the bottom of the water, to the ocean floor, or to any sort of surface, but as adults, they're not capable of movement. Now, movement was an Animalia characteristic that we talked about, but with sponges, this applies to the adult. When they are larvae, they're capable of moving. Water and waste are going to flow out of this very large opening at the top of the sponge called the osculum. Sponges are dependent on a flow of water in order to get their food. Water is going to flow in through intake pores, and it's going to exit this large opening at the top called the osculum. And we'll have a picture up here in just a minute. I'll show you what that looks like. Now, Periphera is the phylum, and it gets its name because there's many pores or openings on the sponge. And if you visualize the brown sea sponge, now we're not talking about your kitchen sponge, but the kind that you see for bath sponges, those are the type of sponges that we're talking about, and they don't have tissues. So if they don't have tissues, they don't have a nervous system, because we know that's ectoderm. These animals do not have that. They don't have digestive, circulatory, or excretory system. That would all come out of mesoderm. And again, there's no mesoderm. There's no tissue formation. So these sponges are totally reliant on the flow of water to come in. They're going to filter out the food or bring the food out of the water, try to trap it. And this is the way they get their oxygen as well. So the outside layer of the sponge is called the pecanoderm, and it's made of what are called little units called pecanocytes. Those are just little units put together to make this overall outside layer. It's not, again, a tissue layer. This is a protective layer for the sponge on the outside, and it is equivalent to the epidermis in animals. Your epidermis is your outer layer of skin, which is a protective barrier for you, and that's the way the pecanoderm is to the sponge. Now the middle layer, because there's going to be an inner layer, middle layer, and outer layer. So the middle layer is called mesophyll. Meso means middle. And in this layer, there are three things: spicules, spongin, and amoebocytes. Now, all sponges, no matter what species, have spicules. Spicules are very sharp, but they don't all have spongin. Some of them have both. Now, what the spicules and spongin do, there's a twofold function for them. They're supportive. There's no skeletal system for a sponge. So this is the only supportive little feature that they have. And because they're very sharp, a lot of times they function for defense, because if something larger is going to try to gobble them up, then they're going to have to get past these spicules and spongin, which may poke them and be very sharp. They also, from what I understand, sort of have a bitter, nasty taste, and therefore the predators are going to be deterred from eating them. So they have a twofold function for defense and for support. Now, amoebocytes are also in the mesophyll, or this middle layer, and the amoebocytes are what digest the food particles. Food is going to have to be filtered out of the water, but there's going to have to be some sort of digestive process in order to get their food. So they rely on these amoebocytes to digest the food. Now, the very inside layer of the sponge is called a layer of collar cells. And the collar cells are working independently. They're not working as a tissue layer. Each cell has got a flagella. And what happens if there's not a current or a flow of water, they beat these flagella back and forth, and that sort of causes a current or a water flow where they can trap the food and bring it in and get it digested into the amoebocyte. So it's not for movement. The flagella is not for the actual movement of the sponge, but it is for movement of the water to make a current in case the water is still, and that's going to bring the food particles in where they can get it digested. Now here's a picture of what the sponge looks like. So on the left, you can see that there's large opening at the top. This is the osculum. And on the side, all of these openings in the sponge, they're really called intake pores. And the intake pores are going to direct the water flow inward into the large cavity in the center, and then all of the waste is going to exit out of the osculum. Now, blowing up this portion over here, it shows you that this inner layer has got all these little teeny tiny cells. They're called collar cells, and each one of them has a flagella, and that flagella is capable of beating back and forth, and it brings it into these amoebocytes. Archaeocytes and amoebocytes are the same thing. I'm going to call them amoebocytes because they typically have a little amoeba type form to them. Also in here, if you can see right here, these are your spicules, and they're pretty sharp. And so this is when, their predators try to bite down on them, they're going to get this sharp spicule in their mouth and hopefully deter them from eating the sponge, because there are a lot of predators to sponges. Starfish move over sponges and eat them. Well, a lot of times starfish will go away if they get a mouthful of these spicules. Then you can see here the amoebocytes down below. They're also for digestion, and on the outside we have these baconocytes it's an epithelial-like cell that together make up the baconoderm.